Good morning, everybody. My topic for today's seminar is Eosinophils in Health and Diseases. History. Eosinophil was discovered in 1874 by Heinrich Caro, director of the German Chemical Company. Paul Ehrlich introduced the term eosinophil in 1879. Introduction. Eosinophils make up about 1 to 6% of white blood cells. They are granulocytes. These are about 12 to 17 micrometer in size. Their half-life is of 8 to 18 hours in bloodstream, and the tissue lifespan estimated as 2 to 5 days. Eosinophils are acid-loving cells that causes them to appear brick red after staining with eosin. They are found in the medulla and the junction between the cortex and medulla of the thymus, and in the lower GIT, ovary, uterus, spleen, and lymph nodes, but not in lungs, skin, esophagus, or some other internal organs under normal condition. They have bilobed nucleus with bright pink colored granulated cytoplasm. Development. Eosinophils develop and mature in bone marrow. They differentiate from myeloid precursor cells in response to cytokine interleukin 3, interleukin 5, and granulocyte macrophage colony stimulating factor. Transcription factors required for eosinophil development are CEBP, GATA1, PU1, FOG. CEMB, if deficient, immature eosinophils are formed. GATA1 contains 2 zinc finger motifs. Its C-terminal is necessary for eosinophil formation. Mechanism by which PU1 induces eosinophil commitment involves downregulation of GATA1 expression. FOG acts as a repressor of eosinophil lineage. Expression of FOG in eosinophil leads to a loss of eosinophil marker and acquisition of multipotent lineage. This is the image showing development of eosinophils. The CD34 positive hematopoietic stem cells convert into eosinophils with the help of transcription factor FOG, GATA1 and PU1. In this image, we can see various stages of eosinophils like promyelocyte, myelocyte, metamyelocyte, band forms of eosinophil. The cytoplasm of eosinophil contains various granules. These granules are crystalloid granule, primary granule, small granules, lipid bodies, and secretory vesicles. Crystalloid granules are electron-dense core surrounded by electron-lucent matrix can take up acidic dyes due to their cationic nature, mostly present in mature eosinophils. Charcot-laden crystal proteins, that is liphospholipase, mostly present in immature eosinophils. Acid phosphatase, aryl sulfatase, catalase, and cytochrome B558 are present in small granules. There are 4 major proteins in granules: major basic protein, eosinophilic cationic protein, eosinophil derived neurotoxin, eosinophil peroxidase. This is the image showing various granules of the eosinophils. This is the another image of crystalloid granule of eosinophil. It shows crystalline core having major basic protein, matrix having eosinophil peroxidase, eosinophil cationic protein, eosinophil derived neurotoxin. Charcot-laden crystal. These are colorless needle-shaped structures. They are designated as galactine 10. They are produced at very high levels. Degranulation mechanism. It is defined as exocytic fusion of granules with plasma membrane during receptor-mediated secretion. In this mechanism, outer leaflet of lipid bilayer membrane surrounding the granule encounters inner leaflet of plasma membrane of the cell. Docking is carried out using SNAP receptors. After docking, granules and plasma membrane fuse together and form reversible structure known as fusion core. Granule contents are subsequently expelled to the exterior of the cell. Piecemeal degranulation. In this mechanism, instead of complete emptying of eosinophil granules, like in cytolytic degranulation, only bits of granule content released in response to stimuli. The remaining part become intact and viable. This takes place in response to interferon gamma and CCL11 to develop cytoplasmic secretory vesicles known as eosinophils sombrero vesicles, which remain viable and fully responsive to subsequent stimuli. This is the image showing various modes of degranulation in eosinophils. These are classical exocytosis, compound exocytosis, piecemeal degranulation, necrosis. The most commonly observed forms of degranulation in allergic diseases are piecemeal degranulation and necrosis. Receptors important for eosinophil function. Interleukin 5 receptor subunit alpha. These are the most prominent cytokine receptor associated with eosinophil. This receptor signaling promotes the development of eosinophils from committed progenitor, induces eosinophil activation and sustain eosinophil survival in peripheral blood and tissues. Chemokine receptors mediate eosinophil chemotaxis in response to eotaxins. Sialic acid glycoprotein like immunoglobulin 8, immunoglobulin lectins 8 and F. These are cell surface immunoglobulin like lectin expressed predominantly by human eosinophils. Antibodies specific to this promote eosinophil apoptosis. So these proteins represent important targets for potential therapeutic ablation. Pattern recognition receptors. These are toll-like receptors localized in endosomes and detect single stranded RNA and is the most prominent toll-like receptors expressed by eosinophils. This is the image showing various types of receptors present on the cell of eosinophils. Interaction of eosinophils with other leukocytes. These eosinophils stimulate T cells to proliferate and produce cytokines in an antigen-specific manner. They regulate the recruitment of Th2 cells in response to allergen sensitization. Eosinophils prime the B cells for antigen-specific IgM production. They also promote the maturation of dendritic cells. They activate neutrophils, causing them to release superoxidase and interleukin 8. This is the image showing various interaction of eosinophils with other leukocytes. Eosinophils and tissue accumulation. Recruitment of eosinophils across the vesicular endothelium occurs in 4 steps. First one is tethering of eosinophils to luminous surface of vascular endothelium. Second one is rolling along the luminous surface of activated endothelium. Third one is firm adhesion to endothelium. And fourth one is transmigration through endothelial cells into target cells. This is the image showing tissue migration of eosinophils. This shows tethering and rolling requires E and P selectins and eotaxins are required for adhesion. Eosinophil primarily is a tissue dwelling cell in human. The tissue eosinophil to blood ratio is 100:1. Eosinophils also home into thymus, mammary gland, the uterus and also controlled by eotaxin 1. Function of eosinophils. They play important role in production of cationic granule proteins, reactive oxygen species, lipid mediators like eicosanoids from leukotrienes and prostaglandins, enzymes such as elastase, growth factors such as transcription growth factor beta, cytokines such as interleukin 1, 2, interleukin 4, interleukin 5, 6, 8, interleukin 13 and TNF alpha. Functions of eosinophils. They also fight helminth colonization, play a role in fighting viral infections, important mediator of allergic responses, and asthma pathogenesis. This is the image showing functions of eosinophils. Eosinophils play an important role in combating diseases like cancer, inflammatory bowel disease, lung diseases, adaptive immunity, allergic, contact dermatitis, transplant rejection by T cell recruitment. Eosinophilia. An increase in eosinophils, that is presence of more than 500 eosinophils per microliter of blood, and is called as eosinophilia. It is present in parasite infection, allergic responses, neoplasm, idiopathic hypereosinophilic syndrome, connective tissue disease. These are some terminologies. Eosinophilia is defined as an absolute eosinophilic count more than 0.5 into 10 to 9 per liter independent of age. Peripheral blood eosinophilia is called mild when it is 0.5 to 1.5 into 10 to 9 per liter. Moderate 1.5 to 5 into 10 to 9 per liter of blood. Marked greater than 5 into 10 to 9 per liter of blood. Persistent eosinophilia is defined as peripheral blood eosinophilia recorded on at least 2 occasions with a minimum time interval of 4 weeks. These are 4 types of primary eosinophilia. Eosinophilia associated with platelet-derived growth factor receptor A, receptor B, PGM1 JAK2 abnormalities. Second one is chronic eosinophilia not otherwise specified. Third one is lymphocytic variant hypereosinophilia, and fourth one is hypereosinophilic syndrome. Absolute eosinophilic count is greater than 600 microliter per microliter of blood in eosinophilia associated with platelet derived growth factor receptor AB variety, and in other varieties of primary eosinophilia, absolute eosinophilic count is greater than 1500 per microliter of blood. Secondary eosinophilia, it is a non-neoplastic most frequent cause of eosinophilia. The causes of secondary eosinophilia are tissue invasive helminths, allergic drugs, metastatic cancers. These are various causes of eosinophilia. Among them, parasitic diseases are most commonly involved in and associated with eosinophilias. Other infections are bacterial, mycobacterial, invasive fungal, rickettsial, yeast, and viral infections. Allergic diseases associated with eosinophilias are allergic rhinitis, atopic dermatitis, urticaria, and asthma. Respiratory tract infections are eosinophilic granulomatous with polyangiitis, chronic eosinophilic pneumonia, bronchiectasis. Skin diseases are bullous pemphigoid, eosinophilic cellulitis. These are parasitic infections associated with eosinophilias. Ascaria, Toxocara canis, filariasis, ancyclostomiasis, strongyloidosis, trichinosis. In filariasis causes tropical pulmonary eosinophilia as a result of migration of adult worms to the lungs. Ancyclostomiasis is one of the main cause of eosinophilia in patients returning from tropical countries. Parasitic infestation. The mechanism of eosinophilia in parasitic disease is thought to be similar to allergic disease, with a Th2 type response mediated by both adaptive Th2 cells and innate lymphoid type 2 cells to helminthic antigens, resulting in increased production of eosinophil, in particular interleukin 5. These are rare causes of eosinophilia. Among them are Churg-Strauss syndrome, eosinophilia myalgia syndrome, Omenn syndrome, hyper IgA syndrome. Eosinophils and asthma. Asthma is a chronic inflammatory disease that is characterized by reversible airway hyperactivity in response to non-specific spasmogenic stimuli. Eosinophils are recruited to lungs and airways by cytokines that are released from activated Th2 cells and by a range of chemokines. This is the image showing how eosinophils is contributed in asthmatic attack. Eosinophils produce leukotrienes, various leukotrienes which cause inflammation, mucus secretion, edema, bronchoconstriction, airway remodeling. Eosinophilic esophagitis. Dysregulated eosinophilia is having central role in pathophysiology of eosinophilic esophagitis. The etiology appears to be dependent on Th2 type cytokine interleukin 5, interleukin 13. This is the image showing eosinophilic esophagitis having eosinophilic infiltration of tissues. Eosinophilic myopathies. It is the most rare, poorly characterized of the eosinophil related disorders. It includes eosinophilic fasciitis, toxic oil syndrome, and eosinophilia myalgia syndrome. Eosinophilic myalgia syndrome is a multi-system disorder. It is defined by US centers as three criteria. First one is peripheral eosinophilia, second one is generalized myalgia, and third one is absence of infectious and neoplastic etiology. This is the image showing eosinophilic myopathies. This is the bone marrow aspirate and bone marrow biopsy image of B lymphoblastic leukemic patient having marked eosinophilia. This is the image showing allergen-induced bronchial hyperactivity and eosinophilic infiltration. This is a filarial worm. The eosinophils are surrounding or attached to a live stunted larvae and eosinophilic material in the surface of the parasite. This is the picture showing allergic rhinitis. Infiltration by eosinophils, mast cells, lymphocytes, and plasma cells, and also a dilatation of capillary vessels are detected. Hypereosinophilic syndromes. It is defined as a disorder of eosinophil hematopoiesis that results in hypereosinophilia greater than 1500 eosinophils per microliter of blood. It is a myeloproliferative disorder associated with damage to multiple organs. This is the image showing hypereosinophilic syndrome. This is a rare case associated with eosinophilia called as Olmsted syndrome. Omenn syndrome, originally described in 1965 with reticular endotheliosis and eosinophilia. It is an inflammatory condition characterized by erythroderma, disquamatus alopecia, chronic diarrhea, failure to thrive, lymphedema, and hepatosplenomegaly. This is the image showing eosinophilic cellulitis called as a Wells disease. This is another image showing hypereosinophilic cellulitis. Pulmonary eosinophilia. Pulmonary eosinophilia is defined as an infiltration of eosinophils into the lung compartment constituting airway, interstitium, and alveoli. Various infections, drugs, parasites, autoimmune processes, malignancies, and obstructive lung diseases have been associated with increased eosinophil in the lungs. This is the image showing pulmonary eosinophilia. Take home message. Eosinophils are clearly capable of more sophisticated immune functions than previously thought, as shown by their non-degranulation responses to distinct stimuli and their complex interaction with other leukocytes and pathogens. A better understanding of different aspects of biology of eosinophils will aid the development of new therapeutic strategies for diseases characterized by eosin degranulation. These are my references. Thank you.